To the second day of Dream, and welcome to all of you who just came today. Many people had to come today, couldn't come yesterday. A few announcements about logistics. There is a poster session that will start a little bit later today. The poster session, you can start putting up your posters at about 10. And it's in the one floor upstairs, so please take your posters and go there. There will be also a posting board there where you can post your jobs or post your wishes or whatever, or meeting people, whatever. Please feel free to use it for communication. Okay, let's start with the day. It's my great honor to introduce to you Leslie Vosshall. Leslie is the Robin Chambers Neustein Professor and Head of Lab of Neurogenetics at the Rockefeller University, where she leads the Neurogenetics and Behavior Lab. And in part because of that is that we invited here, she was very generous to give, and along with Andreas Keller, who will be the next speaker after her, the data for the Olfaction Prediction Dream Challenge. Leslie's interest is in molecular neurobiology, and her research program aims to understand the genetics basis of behavior, with particular emphasis on how organisms perceive and respond to external sensory stimuli, and how these responses are modulated by internal physiological states. She is an investigator of the HHMI, and this year was selected to the National Academy of Sciences, so congratulations on that. It's your floor. Thank you.
Hi, good morning. It's a great pleasure to meet Gustavo, I think for the first time. And to be here, sort of the culmination of the world's greatest attempt for Andreas Keller and me to have you guys do our homework for us, right? It's an amazing thing that the Dream Challenge was able to do. I don't have the numbers right now, but Andreas collected all the data, and you'll hear some of the details of the data when I finish my talk in about 30 minutes. And so when we put the challenge together with Pablo and Guillermo, we thought the most likely thing is we'll put it up and it will just be ignored. We'll just sit there and watch the wiki fill up with nothing. But in fact, some huge number of people actually were interested in this coding problem of how you take a molecule, a small chemical volatile, and turn it into a percept. So we're really excited. I'd like to congratulate the winners who are here today, and also all the participants. It's not a contest, just two people are playing. I should also disclose that for the last few years, I've been a member of the Scientific Advisory Board of IFF, so a company that's really interested in this problem. And it's been a lot of fun to smell odors when I visit them, and it's great to have bridge between academic science and industry. So this is a slide that Andreas may also show. But if we think about all of the different senses that we have, olfaction has fascinated people for obviously thousands of years. There has been no satisfactory explanation of how you go from these small molecules to a particular percept. So we all know that in the world there's some number of odorants that we encounter every day when we eat, when we walk around the world, and I would say even in spite of the great efforts of the Dream Challenge, we still have no clear idea about how this works. So my goal today is to walk you through just a basic introduction of what is the sense of smell, what is the coding potential that's conferred by the proteins that detect odors, the coding potential conferred by the brain circuits that process that information, and what the prospects, if any, are for us to figure this out. So here's the five senses, and I would say, again, that olfaction, I wouldn't say it's pathetic what we know about it, but it's pathetic what we know about it. So if we go to vision, we know exactly what the range and resolution of the visual system is. We know that we can't see UV and we can't see IR. So we know exactly the boundaries of the wavelengths of what humans can detect. We also know that there's a linear relationship between the wavelength of a light and the color that's perceived. So I could write a nanometer wavelength, and those of you with great knowledge of color perception, or with a computer could tell me it's blue, it's violet, it's red. So we know exactly how you translate the stimulus into the percept. And we know enormous information about how these stimuli are detected and turned into a percept for vision. Now, audition is the same thing. We have a linear segment of information that takes frequency information and turns it into low tones, high frequency tones, low tones, and that's how you're listening to me right now, just a simple stimulus code that leads to a percept. And again, we know what the resolution of the human auditory system is, and we know what the range is. We are unable to hear very ultrasonic vocalizations produced by, let's say, mice, and we're not able to detect the very, very low frequency that other animals use to communicate. So the problem of the stimulus percept has effectively been solved for these two modalities. Moving onward, for touch, again, you can make a simple array of the amount of pressure that you use to deflect surfaces will give you a linear readout of how painful it is, right? So that's also relatively clear. The chemical senses have been much harder because it's hard to figure out how do you translate these molecules into any kind of a dimensionality from left to right, from violet to blue to red. For taste, those of you who've been eating this morning, you can divide it into sort of simple taste qualities that are discrete in the percept and that have a relatively narrow set of molecules. So you have sugars that will activate the sweet pathway, actually a large number of bitters that will activate the bitter pathway, sours, umami, and salt. So taste is pretty discrete. We know sort of a lot about the coding potential of the gustatory system. And smell remains something that's been, as I said, quite mysterious and has been studied and written about for thousands of years. We had a bump of interest in 1991 when Bach and Axel identified the proteins that mediate our detection of molecules, our perception of them into the brain. So I'll just pause for a second and tell you guys what the olfactory system can do, what different kinds of metrics that when we smell things, are able to take in. And this relates directly to the challenge. So in the data that Andreas Keller will tell you about, we asked subjects to give us information along several different axes. So valence is just a fancy word for pleasant or unpleasant. And so this is the dead horse arum plant, which most people find really unpleasant. It smells like rotting meat. And this is a rose, which most people find very pleasant. And so these produce different molecules, many, many hundreds of different molecules, some of which in combination will produce, for most people, a percept of pleasant and very unpleasant. And so this is how we measure these things in the laboratory. So this is a picture from one of Andreas's early smell studies, where the tools of the trade are quite simple. So you have a human being. You have a test administrator, you have a barcode reader. The human being picks up one of these odor vials, scans the barcode, opens the lid, sniffs, and answers a question. How pleasant is it? How unpleasant is it? How strong is it? And then if you see in the background all these different descriptors, dead horse, coffee, urine, lemon. And so we have a sort of a poverty of words that can describe the kinds of things that this person is smelling. But valence, everybody gets that, disgusting or beautiful, right? And so these valence you can put onto a single axis from extremely disgusting to extremely beautiful. And so all of the data that went into the Dream Challenge was collected under these conditions: a human being, odor vials, and a computer to collect the opinion of the person smelling the odor. And so I shouldn't say that rose is a rose, right? So red, most people, if you can see red, you'll assume that red is red. Olfaction has all of these cultural layers on top, so that depending on where you are in the world, cilantro will be really great or really disgusting. Durian will be really great in Asia, but really disgusting to us. French cheese will be really disgusting to people around the world, but perhaps favored by people who like blue cheese. So layered on top of the chemistry, to the proteins, to the circuits, is also culture. And culture enriches and contaminates every single piece of data that went into the dream challenge. We can't get someone and disambiguate them from their past experience of these odors. So I talked about valence. Do you like it? Do you not like it? Another very important piece of information that the olfactory system gives you is intensity. How strong is it? How weak is it? And people differ greatly in how sensitive they are to odor. So something that will appear to be really strong to one person will be barely detectable or not detectable by another. And it usually just scales with the number of molecules. So one rose is a lot less smelly than many roses. Okay, so another area is categorization. So we have all of these different things out in the world. It's easy to do this with these pictures, right? You have fish, and you can decide what your valence is here, right? Fish, you have citrus, chocolate, sandalwood, cinnamon, tobacco, roses, violets, some kind of lily, some kind of another lily, and grass. And so you could take these pictures and categorize them really simply. And so you could take the florals and say, like, these all sort of smell like flowers, right? So all of these flowers smell very different from these non-flower stimuli. And so you can simplify the dimensions of the stimulus from the 6 or 7 that are shown on this slide, and then simplify them down to a single category of floral, right? Nevertheless, unless you have a severe cold or a severe deficit in your ability to smell, most people will be able to discriminate the scents of these flowers quite readily. And the categories are very heavily governed by our experience, right? So many of the things that we have categories for were established by the flavor and fragrance industry. They generally relate to things that we produce or consume. And so again, we have a poverty of words to describe the available smells in the world. If you went around and smelled everything in Philadelphia today, you would very rapidly run out of words to describe the individual things that you're smelling if you want to go beyond just the kind of cliched list of descriptors that we have. So even though the Olfactory Dream Challenge had categories in it, I think Andreas and I share the strong feeling that words really fail us when we come to smell. So words generally are pretty useless. They're not completely useless because you can categorize things somewhat, but they're heavily influenced by a cultural overlay, and the categories will change radically when you go to different parts of the world. So, and that's much less of a problem in audition or vision, where a particular frequency of sound or color generally is not associated with a particular emotional valence. It's a stimulus. And then lastly is this point of discrimination. I kind of foreshadow this, that although you can do a really big dimensionality reduction and take all odors and put them into a floral bucket, a rose is not a rose is not a rose, because here's a stock photo of a bouquet of roses. So people around the world are constantly breeding different varietals of roses that they're all rose, they all have the canonical molecules that make them smell like roses, but if you line them up and close your eyes, they're all readily discriminable. I would argue even if you happen to grow roses and you have a plant in your yard that has 20, if you're lucky, 20 different blossoms, if you go from blossom to blossom to blossom, they will all smell slightly different because they're at different stages of ripening, whatever the word is, and or decay. So, and this is a great thing. It's a great capacity of the olfactory system to go past pleasant to unpleasant, intense to not intense, to different categories, to a really rich set of abilities of us to tell different things apart. And so Noam Sobel did this great experiment that we amplified on last year, which is to abstract all these concepts of is it intense, is it not intense, is it disgusting, or is it beautiful, by just mixing a lot of stuff together, just a lot of random stuff together, which perfumers would never do, right? I think Andreas and I showed these mixtures to Frederick Mahl, who's a perfumer, makes beautiful fragrances. The last thing a perfumer would do is just take a bunch of crap off the shelf and mix it together. And what happens when you mix things together, as Noam Sobel's group did here, is that it just starts to smell like crap, right? It loses any kind of an emotional valence to anything that you can tether into the world. And so he could show that as you mix more and more substances together, it sort of starts to get this quality of white noise, some sort of which they call olfactory white. So you don't even need to use words because it doesn't smell like anything. You can detect a smell, and but you can't put a word to it, which is a genius experiment because then you can start really testing how good are people at discriminating things independent of this problem of culture and categorization. And there's too many data points here, but as he starts mixing lots of these molecules together, and initially with 2 or 3, it might smell like something you could put a word to. As you mix in 10, 15, 20 to 30 different things, they really all sort of start smelling the same. But if you look at the accuracy of discrimination and the confidence with which people discriminate, the more you mix together, the harder it is for people to tell them apart. Nevertheless, the accuracy to discriminate them is well above chance. So a little bit of a taste of how good people are at discriminating different odors. And I'm now going to show a slide from the most hated and most controversial paper we ever published, which is taking these olfactory whites and making many of these mixtures of 10 random compounds, 20 random compounds, or 30 random compounds, and then asking, using that smell testing paradigm, can people tell them apart, right? So let's go to the mixture of 30. So if you have 30 different molecules and then 30 other different molecules that don't overlap, and you ask how many of these individual people can tell them apart from chance. So it's like a shell game. You have 1 red vial and 2 blue vials. Tell me which one is different. And so all of these people are able to tell the difference when they're completely different. Then you start making them more and more similar. So now you have only about 2/3 of the molecules are different. Still, people do really, really well. And even in the case where you have 29 molecules are identical, but there's one different, some fraction of the people, these individuals, are actually able to pick out that one difference above chance. So we think the discriminative capacity of humans is really impressive. And in these experiments, we can abstract it away from the categories that contaminate our ability to really look at the human sense of smell. So this is the problem that the Dream Olfaction Challenge sought to solve, is how do you go from a single odor molecule to a percept? So unless you happen to be an expert, I don't know if there's experts in the room, chemists can, if you're not a chemist, you can see shapes, right? So there's a convention by which chemical structures are drawn, so anybody can see the shapes are different. If you're a chemist, you can see that there's nothing exotic here. Most olfactory molecules, with the exception of the sulfurs that were in the challenge, are composed of a small number of different atoms. And all of these are very small molecular weight, because in order to smell something, it has to be volatile. Large molecules that don't go into the air, by definition, are odorless, so you need to be able to inhale it. So again, unless you're an expert, you have no way of saying which one of these is like a cedar sandalwood, which one is like grass, which one is like violets, and which one is like roses. And so this is the answer. This is ISO-E super, which is a kind of a cedarwood, cis-3-hexanol, which is a grass beta-ionone, and I think this was a rose oxide. So again, by trial and error, people make this stuff. It's like, wow, cis-3-hexanol smells exactly like cut grass, right? People have been busy since there's basically no sandalwood trees left in the world. They've all been cut down. People are very interested in making new sandalwood essences, and it's a huge struggle. You can find different companies have made these different variants of it. Each of them smells slightly different, and it's very difficult to figure out how you go from these structures to some sort of a category of cedarwood or sandalwood. Making the coding problem even more interesting is that most things that we smell outside the lab, outside of the study that people did, are not single molecules. So when you smell one of the roses that I showed you, if you separate out the volatile molecules coming off the rose, you get hundreds of them present at different concentrations. And so what the brain has to do is take in the information about which molecules are there, what the concentration is, and then somehow pull that together into a code that indicates not only the category, which is rose, the concentration, which is the intensity, and the valence, do I like it or do I not like it? So the olfactory system has a big job. And again, on this problem of structure percept, each of these molecules is used both in the laboratory and in industry to encode the scent of a rose. And if you were to sit down and try to sketch, let me make the next big rose molecule, it's not so clear how you would do it with great accuracy. There are some structural similarities, but there's no obvious route to clone these and make a new rose molecule. And as I mentioned, these substances work in combination. So if you take sort of the greatest hits of different molecules found in these different flowers, so each of these flowers, from roses to violets to hyacinth to chrysanthemum, they all are obviously discriminable. Underlying this, though, is a lot of overlap in the molecules. So beta-ionone is found in many molecules, and eugenol is found in many flowers. So again, the brain is able to take all this information and parse it out. So I'm going to go into a little bit about the neural circuits of how the brain does this. Then I'm going to go a little bit into the discovery of the odorant receptors that detects all of these molecules. And then I'll close with what little we know about the differences between different receptors that encode different percepts. So we have to always start with human anatomy. So we all have noses. The information from odorants has to reach this tiny strip of olfactory epithelium here in the posterior dorsal nose. And so the molecules go up there and stimulate neural activity that goes to the olfactory bulb. And if you zoom in on that little strip, you find millions and millions of olfactory neurons that I'll show you in a minute, each express a different flavor of odorant receptor. So you have a set of red neurons, a set of green neurons, a set of blue neurons, and there's this magical wiring phenomenon that puts all of the information together into these clusters called olfactory glomeruli. So you sort of have a nexus of information. The green receptor collects all the information from the green receptors, and this code, we believe, is subsequently read by the brain to encode the percept of an odor. And so Andreas and I commissioned this movie a few years ago. Imagine these are odor molecules flying around in the world. And they've gone into your nose, and they begin to interact with these olfactory neurons that are have different chemical specificities. So if we zoom in on the cartoon version of the olfactory neurons, you're sniffing, and let's say this blue odorant is coming along, and it docks onto this receptor and begins to make activity. It doesn't fit into the green receptor, so there's no activity. The red receptor is very strongly activated by this odorant. So you can see a single odorant activates multiple different types of olfactory neurons. This is the first relay center in the brain, and so you can see that a code starts to emerge. A strong activation here, medium activation here, weak activation here, no activation there encodes the blue odor, right? It's a simplistic view of how olfaction works, but elements are almost certainly correct. So the idea is that you start putting a single molecule across an array of different receptors to yield a combinatorial code that elicits the percept of the odor. And so this is now really old work. Some of the early attempts to take the wiring diagram of the olfactory system and relate it to a functional diagram. So work from Ben Rubin and Larry Katz. If you zoom in on a little part of the brain in a rodent, and most of the work that we know about olfaction is actually based on work in rodents. But we have reason to believe that the basic biology is the same in humans. So you can do this intrinsic signal imaging, which when you ask these animals to sniff an odor, you can look at patterns of activation in their olfactory bulbs. And these little blobs of dark color are different glomeruli lighting up when a rat has smelled amyl acetate. So amyl acetate will activate 6 or 7 of these glomeruli. Peanut butter, complex odor will activate these too. And the take home here is that different molecules are activating different combinations of glomeruli. It isn't that a single odor activates a single glomerulus, because that would give you a very limited coding potential, because mice have only about 1 or 2,000 of these glomeruli. So we think that the activation of these glomeruli gives you something of an identity code, which comes down to categorization and identity. We also have to encode concentration. And these and other experiments have shown really beautifully that, again, if you're imaging in the olfactory bulb, which is here, you put on essentially just clean air, you get no activation. As you increase the concentration of this odorant, you activate one glomeruli, then two, then three, and at higher concentrations, you activate many glomeruli. So this is a really simple way to encode concentration. The more of these molecules you have, the more of these glomeruli you start blinking on. So how does information get from the olfactory bulb to higher brain? If we just handed this information forward, you would have very limited coding potential, right? So if you have 1,000 glomeruli in the mouse and they all light up, at some point you have to interpret that information and put it together with the whatever cultural underpinning or whatever learning you have. And so this foundational work from Richard Axel showed that information is then, it's decomposed in the piriform cortex, which is sort of the early part of the brain that processes odors. So you could view the receptors as taking in information about the molecule that's being smelled, the olfactory bulb representing it in this two-dimensional map, and then the piriform cortex decomposing the information into patterns of activation. So this is the olfactory bulb here. So this is a rodent, so the nose is out here. This is the olfactory bulb where you get the first relay of information. Information streams back here to piriform cortex. So what Dan Stetler did was just look down at the brain of the rodent while it was sniffing different molecules to see what kinds of patterns of information emerge. You could imagine that if you have let's say an octanol spot in the piriform cortex, then you would have very localized activity. And that's absolutely not the case. So if you look at a little part of the piriform cortex, just with solvent, you get no cells activated. Acetophenone, you get a single cell activated. Benzaldehyde, a single different cell activated. Octanol, a whole cluster of cells. Mouse urine, which mice really love to smell, it has strong social context for them, activates different cells. And this thiol, which also is socially active, activates a different set of cells. So there's no regionalization. It isn't like there's an acetophenone part of the piriform cortex or an octanol part of the piriform cortex. And again, the piriform cortex has a way of encoding concentration, this really important idea of what am I smelling, how intense is it, and what does it mean to me? So imaging the same region in the same animal of piriform cortex, as you go from 3 parts per million, you activate this ensemble of cells. You go up to 16, you sort of double, and you go up to 62 and you increase it further. So the more molecules of odorant you have, the more of these piriform cortex cells you recruit. And here's this beautiful assembly of what the brain is doing with these odors. It's not putting them into distinct cartridges of odor identity. It's distributing the information over a wide group of cells. So it's a huge substrate for encoding odors. So again, looking from across this whole arc of piriform cortex, you can see that essentially random groups of neurons are being activated. And these maps change across different animals, so every animal will display the information differently. And the circuit is highly plastic, so you can, meaning it's a great way to write memory. So if you activate this set of cells and you shock the animal, it will always remember that pinene is bad. If you synthetically activate this same set of piriform cortex neurons, they'll be terrified, right? So it's a beautiful substrate, very information rich, to be able to encode a very large number of odors and odor experiences. And so that's summarized here in this really nice cartoon where there's great order in the olfactory epithelium. Each of these dots represents a different neuron expressing a single receptor. As I mentioned, these go into these nodes, sort of simple nodes of information. Each node is a particular receptor. And then it just gets distributed in a really complicated and random way into the piriform cortex. So it's an incredibly efficient way to maximize information processing because it's like a rewritable disk. You don't have any fixed address for any particular odor or any odor experience. So how do we get all of these odor experiences? Each of these, again, is a different molecule, and the olfaction challenge winner should be able to tell me what each of these, how it's perceived with intensity and valence. So the question is, how do we build sensors for all of these different odors? Keeping in mind that we can do an enormous amount just with 3 visual receptors, so you can see the entire visual spectrum just by combinatorial activation of 3 visual receptors. And as I mentioned at the beginning, the problem is very confusing and complicated because we don't have just a linear array of light wavelength color interactions. All of these crazily different molecules have a different scent and a different percept. How do you capture them all? So early work from Odorant Lancet suggested some biochemical activity in the nose, that there must be something related to G protein coupled signaling. So the same kind of signaling that we're using right now to make our visual system work seems to operate in the nose. And this was a mystery for a couple of decades when people were actively looking for these proteins. And so this is what a G protein coupled receptor looks like. You have a molecule that binds a receptor, and the binding of the receptor will trigger a signal transduction cascade that leads to the depolarization of the neuron. So these were the kinds of proteins that people were looking for, with this idea that, okay, you have to encode all these different odors. How do you do it? Do you do it like the visual system, where you have a very small number and then you just sort it out later and have some information transfer function, or do you make a lot of receptors? And so this was solved now in 1991 by Richard Axel and Linda Bach, who found a very large family of proteins like this. So this is a cartoon of a G protein coupled receptor with an N terminus outside, a C terminus inside. And these are the proteins that have some capacity to interact with odorants and confer odor specificity and odor valence in the brain. So the question arises, how do we use these things to smell everything, right? If you have only 400 receptors and each binds a single molecule, humans can smell 400 molecules, and we know that's not the case. I could make you guys start writing lists of all the things you could smell. Collectively, you would, I hope, rapidly exceed 400. What's also interesting about humans is that our genome is cluttered with odorant receptors, so we have a lot of them. About half of them have been lost in the process of evolution. From our vertebrate ancestors, common ancestors. So humans have about 400 of these proteins, each with variation in amino acids that's concentrated in these regions, in these transmembrane regions. So the way that we think this works is that you have this sort of blank slate of 400 odorant receptors that each is interacting with some subset of odorants. And if we use an array of these little circles as a sort of a model of the odorant receptors, at rest, if you're in an odor-free room, like this room is totally odor-free, you might imagine a simple odor like vanilla is going to activate a few of these receptors strongly, a few receptors weakly, and then even others very weakly. And so somehow the brain is abstracting this into a code for vanilla. Jasmine, which is a really complex odor, you might imagine at low concentrations it's activating a large ensemble of neurons, but again, because each is activated to a different extent, you can abstract this as jasmine. And then if you sort of double, triple the concentration of jasmine, all of a sudden you get a very strong percept that can sort of go over into unpleasant valence because you're activating additional receptors and additional circuits. There's also really clear evidence of interactions between odorants, which complicates any attempt to make a link between stimulus and percept. So bourgeonal, which is lily of the valley, activates a small number of receptors. Undecanal, which is an aldehyde that smells like citrus, activates others. But if you mix them together, you will suppress all of this activity, and you'll just be left with these two. So there's these strong interactions that affect how mixtures are perceived by humans. And so when the receptors were cloned and people began to ask what kinds of odor ligands activate these receptors, all of this coding was immediately clear. So this is a beautiful paper from Hiro Matsunami with Joel Mainland when he was still at Duke. He's now at Monell. And so what's arrayed here is all of these different smell molecules and all of these different receptors. And what you can see right away is this receptor responds to many of the odorants. This receptor responds to one odorant. So receptors differ in how promiscuous they are. There's some that are very promiscuous, one that are very specific. And what's clear is that a particular odorant will activate multiple receptors, and a particular receptor will respond to lots of odors. So it's a massive combinatorial code. So just with 400 receptors, you can encode an enormous amount of information, layering on top this fact that you have odor mixture interactions, positive or negative. So it's kind of an unprecedented information array in the brain. And so each of us sitting in the room has a different experience of the world. This was made really clear by Idan Menashe in Durand Lancet's lab, where they sequenced odorant receptors from 91 human subjects and asked which ones are functional in green, which one have only one functional copy, and which are non-functional. And you can see this is like a DNA fingerprint. Each of these people has a completely different array of these 26 receptors. Some have a functional copy where someone else has a non-functional copy. And so these differences between different people is most likely the explanation for something called specific anosmia, where some people are able to smell scents that others are not. And so a classic case of this is these two sex steroid-derived odors that are volatile. So testosterone is non-volatile, but you do some chemistry and these are quite volatile. And if you ask people in our study and elsewhere what they smell like, some people find them incredibly nasty, so negative valence. They'll use descriptors like sweaty and urinous. Other people will give it sort of positive valence and will call it sweet and floral. And somewhere between 0.5 and 10% of people claim that the vial is completely empty. So it's an incredible range of difference of opinion of what these molecules smell like. And so this is from our smell study. If you give, again, that list of descriptors to people, there's an incredible consensus that vanillin, which is artificial vanilla, smells like vanilla. So people are good at, they remember that vanilla corresponds to this semantic percept. Giving people this list of 141 descriptors, they really use very few for vanilla. There's a general agreement what vanilla smells like. Androstenone is all over the place. Again, depending on whether you find it floral, let me find some good ones, floral or fragrant versus chemical, musky, cleaning fluid, dead animal, right? So people don't really know what to do with this odorant. It's sort of unfamiliar, and there's an enormous difference in how people perceive it. And so this big breakthrough to try to figure out what is the cause for all of these differences was inspired by colorblindness. So if you're colorblind, you know that you have a genetic defect in one of your visual receptors. And so Matsunami, this is a few years ago, teamed up with Andreas in my lab to try to find odorant receptors that would interact with this molecule. And so you take the receptors out of the human and you put them into cell lines that will be able to couple the activation of the receptor to a luciferase signal. And so doing this, screening essentially all of the human odorant receptors against androstenone, this odor that people perceive as dead animal, floral, or pleasant. Hiro and Hanyi Zhuang found a single receptor called OR74 that's highly activated by androstenone. And then furthermore, profiling all of these different odors, Matsunami found that this receptor responds only to these two sex steroid-derived odors. So it's a case, this is a really specific receptor. I showed you from Mainland and Matsunami's work that some receptors are really promiscuous. They'll respond to dozens of odorants. Here's a highly selective receptor. And so we reason that if you have some sort of a genetic defect in this receptor, it might affect your ability to find androstenone disgusting or find androstenone high intensity. And so what we found is that in red are subjects carrying nucleotide polymorphisms that make the receptor inactive. So this is the Wildtype receptor, gives strong activation to androstenone and androstedienone. People walking around with a defective allele don't respond to either of these. And in subsequent studies, two different groups, one at Monell and one, I think, in Norway, found that, again, people carrying non-functional alleles of this receptor found this molecule not very intense, whereas people with normal copies of the receptor found it very intense. So it's these two amino acids that alone are able to completely destroy the activity of this receptor, which is pretty cool, right? There's almost 400 amino acids here, and just two will completely detune the receptor. And so that's shown pharmacologically here. So these are cells expressing this receptor, stimulated with either androstenone or androstadienone. This is what a normal person's receptor will do. So it's pretty sensitive and it gives high activation. In red is a person who has both of those amino acids mutated, and synthetically, either one of the amino acids mutated. So just a single amino acid will drastically reduce the activity of this receptor. Mutating both of them effectively kills the receptor. So what Andreas did was basically ask, what happens if you're walking around with one defective receptor? And had our subjects sniff this whole panel of different odorants, and then ask, for each of these odorants, which is significantly affected by mutations in this receptor OR74. And exactly like the highly specific tuning of the receptor, he found that the only two molecules that were affected were androstenone and androstadienone. So these things are highly correlated with unpleasantness. So if you are missing one copy of the gene, you will find androstenone to be more vanilla and less sickening than a person who has two functional copies. So it's a really clear demonstration, both on valence and here on intensity, that these two mutations in a single receptor are highly correlated with the perception of this molecule. And here's the other missing slide showing that another group, again, confirmed these findings. We always sleep better if someone independently in a completely different cohort of people, again, finds that people with two functional copies find androstenone very intense, and then if you're missing one or both copies, intensity drops. So this was cool, showing that the variation in perception really can be mapped, in this case, to a single receptor. So what do we know about the structure percept problem at the level of the receptor? So I showed you this indirect evidence going from mutations in the receptor to the percept, that these two amino acids are heavily contributing to the percept. A number of people have struggled both with experimental approaches and with modeling approaches to try to figure out where the odorants are binding. Where do all of the molecules that smell like rose, where in this protein are they binding? And there's reasonable evidence, although it's far from solved, that in these regions that sit deep in the membrane, in the olfactory neuron, that there's residues here that if you mutate them, you abrogate the tuning of the receptor. I think this is one of the biggest unsolved problems in the biochemistry of smell, is how do you go in these 400 receptors that all have a lot of variable residues here to figuring out how they're binding iso-E-super or cis-3-hexanol to be able to give the percept of these molecules. Here's a modeling approach that comes up with basically the same answer, that these two different odorants, nonanol and octanol, are binding, again, in this region between transmembrane domains 5 and 6. Now, if we had a really good model of where these odorants were binding, I think we would get much closer to a transformation from percept, from structure to percept. So I'm going to close with what we do know and what we don't know. So we know that there are hundreds of odorant receptors expressed in the nose that have combinatorial encoding of odors. We know that these are represented initially in an ordered way and then ultimately in a disordered way to encode a vast number of different olfactory percepts in the brain. There's a lot of things that we don't know that it's going to be a huge challenge to try to figure them out, and it really comes down to dimensionality, right? We know that vision and audition, the stimulus varies across this sort of linear array. The receptors are few and well understood. In olfaction, we only know the stimulus tuning of the receptors for about 5 to 10% of all receptors, so we still have 90 to 95% of them where we don't have any idea what they respond to. This is a sort of a map of physical chemical space. We've only sort of sampled a very small number of possible molecules. And the perceptual space is something that's also probably incorrect, where people have categorized perceptual space into floral and rosy and woody. So we don't really know the dimensions of the receptors or the stimuli or the percept. And the question of how many of these molecules there are, I think, is one of the biggest problems. So we just have no idea how many different scent molecules there are. You know, if you go into any retail store, you're not going to encounter more than a couple hundred scent molecules. If you go into a restaurant, encounter a couple hundred scent molecules. But the only thing that a molecule has to do to have a perceived odor is to be of a molecular weight under 500 and have a few simple atoms. And so this cheminformatics group trying to figure out for pharmacology purposes, how many different molecules are there that are druggable, that would fit into a drug discovery screen, and got this frightening number of 166 billion molecules in theory can be made, right? So, and so we have no idea how many of these are, give a conscious olfactory percept. We have no idea how the receptors would deal with this vast amount of information. And I look forward in the following talks to you guys telling me how 166 billion odors are represented. Thank you.
Thanks, that was great. I just had a couple quick questions. One of them, you gave the example of the orange and the lily of the valley and how they shut down one receptor. Is the mechanism for that understood?
So we think the mechanism, it comes down to G-protein-coupled receptor pharmacology. So we know G-protein-coupled receptors, of which the human odorant receptors are a member, are subject to agonism and antagonism relationships. So that's well known for something like the beta-adrenergic receptor, where you have a binding site, and then you either have a competitive binding that goes into the site and knocks the ligand out, or a non-competitive binding site. So we know the pharmacology for something like beta-adrenergic. That specific example, because we don't know where any of those things bind, can't at the moment tell you if it's competitive or non-competitive. But the idea is that these molecules are interacting somewhere within the inner leaflet of the membrane on that receptor to shut it down, right?
I see. And then the second question was just, there was one of those slides that you said this is our most hated paper.
Oh, yes.
What was the deal? That seemed great.
So we have an expert in the room that we've had a lot of conversations with. So the idea is, how many odors can you discriminate, right? So if I take the catalog of any of the flavor and fragrance companies, which will have about 10,000 molecules, and I want to figure out how many of them can we tell apart, right? We get Andreas and Peggy does all our smell studies. We just get people in and we just make pairwise combinations. That's a lot of odorants, right? So if there's 166 billion molecules that could be odorants, it's a lot of things to test. So what we did is to kind of sharpen the question, is to make these Noem-Sobel mixtures where we just start mixing a lot of stuff together with the idea as you get more and more and more overlap, it should be harder and harder and harder for people to tell the difference. And that's absolutely true. And then we did an extrapolation to try to figure out, if you took all such mixtures that we can make, given the palette that we were playing with, how many of those should people be able to discriminate? And so the answer that we came up with is really large, 1 trillion. And so the question is, was that extrapolation correct? So that's the question. That's why it's the most hated paper, because some people think that there's, number one, how do we know that mixing all those odors together will actually lead to molecules that smell different, because you're sampling? You're sampling from 10 to the 29th combinations. Maybe they start to all be the same. And then number two, how do you make an extrapolation that's that big without making a huge error?
Sure. I have just a short question, which is the interpretation of the sense of smell and actually sensing it to independent phenomena? Because you mentioned cultural effect have a big influence on how we interpret these senses. So I was wondering if there is the whole relationship with age based on what we learn, and if there's any study that's like basically look at how when we are younger perceive the smell versus when we have more experience from the word, how does it affect the interpretation of the smell?
So almost all of our opinions about smell are learned. So I think that there's a little bit of data that young babies find all smells really interesting, so they'll attend to them, and they only later, early in life, learn that that's disgusting, like excrement's disgusting, this is good, that's bad. So I think humans are contaminated by culture very early in life.
Okay, thank you very much.